Start.
Okay, thank you. So I'm a postdoc working with UHI at IBM Research, and I'm going to tell you about some work we've been doing recently studying the role of energy dissipation in chemotaxis signaling. So I'll start with kind of an overview of the chemosensory network in E. coli. E. coli has thousands of receptors on the cell surface, and these can bind to various extracellular ligands, and this modifies the phosphorylation of a messenger protein called QY that carries this message to the cell motors. So in this way, the bacteria responds to its environment and can decide whether to swim forward or tumble and change direction with the aim of biasing its motion up concentration gradients. So you can think of this as just a simple input-output signaling circuit. And there's also an adaptive mechanism on top of this that I want to mention. The cell can methylate or demethylate these receptors. And the effect of this is it modifies the phosphorylation reaction and shifts the downstream response across a wide range of concentrations. So this allows the cell to maintain sensitivity to its current environment. And we'll see an example of what that looks like later in the talk. So there's two key aspects of the system I want to focus on. The first is the energy dissipation. This phosphorylation reaction is driven out of equilibrium by ATP hydrolysis, supplying energy to the system. And the second is that these receptors aren't isolated on the cell surface. They form this extended cooperative array with cooperative coupling between neighboring receptors. So I'll touch on both of these throughout the talk. Before getting into this, I want to give a little more background and tell you about the kind of old classic model used to understand the behavior of the system that's seen a lot of success describing various experimental behaviors, but ignores both the dissipation and the spatial structure I just mentioned. And this is the so-called MWC model. So here I've written a simple Hamiltonian formulation of it. It involves a cluster of N receptors, and each of these receptors have binding or occupancy degrees of freedom. They can either be bound to ligand or not bound. And then the other key assumption of this model is that there's an all or none activity behavior within this cluster. So all the receptors are either simultaneously active or inactive, and that's captured by this degree of freedom S. So we have a simple Hamiltonian with binary degrees of freedom and a few energy parameters. There's a chemical potential-like term that depends on the ligand concentration. There's coupling between activity and binding, and there's some energy difference between the active and inactive state in the absence of binding. And then, of course, we can use Boltzmann statistics to derive things like expectation values for the activity as a function of ligand concentration. So I'll give you one example where this has been successful, but there's a pretty broad literature using this model to describe various experimental behaviors of this chemotaxis system. Here's some data from Howard Berg's lab, and they use FRET, which is a fluorescence imaging technique, to measure the amount of phosphorylated protein inside the cell as a function of the external ligand concentration. And they repeat this measurement for different mutants of E. coli, where they fix the methylation level. Now remember, methylation, I told you, was this mechanism for adaptation. So what changing methylation does is it shifts these response curves across a wide range of ligand concentration. And to capture all this behavior with the MWC model, all they had to assume was that one of these energy parameters depends on methylation. And in fact, they could assume a linear dependence. So then this MWC model has 4 or 5 parameters, and they can simultaneously fit all of this response data with the same set of parameters. Okay, so as I said, this is just one example, but there's a broad literature using this model to describe the system. But now I want to tell you about 2 experiments and some modeling we've done that goes beyond this MWC model. These are measurements that show the inherent dissipative nature of the system and give some insight into the underlying microscopic mechanisms for signaling. Okay, so this first measurement comes out of Jerry Hazelbauer's lab, and they measured both the input and output side of the signaling circuit, the amount of phosphorylated protein and the binding to the receptors as a function of ligand concentration. So these top plots are showing exactly what I showed you earlier. It's the amount of phosphorylated protein as a function of ligand concentration for different E. coli mutants with fixed methylation level. And again, we see that methylation shifts the response across a wide range of concentrations. But the surprising result from this paper is on the input side of the circuit, the binding response curves hardly depend at all on methylation. So here I've overlaid them, and you can see that there's a big disproportionality between the adaptive shift in the downstream response compared to the uh, input response. And the claim is that this is an inherently non-equilibrium behavior. So I'll give you an example using this MWC model, but I'll point you to our paper for kind of more general and broad arguments about this. Um, so here I've fit the MWC model to this QIP, the downstream response of the system, and the model makes a prediction for the binding response curves, which are shown here by the lines. And you'll see that they spread across a wide range of ligand concentrations, which is contradictory to the experimental data I just showed you. And the basic intuition behind what's going on here is that this is an equilibrium model with reciprocal interactions. So if we modify this energy dramatically to shift these curves across such a wide range, this feeds back through the interaction term to the binding degrees of freedom and spreads those curves across a similarly wide range. So what we've done, we've kind of built the simplest possible non-equilibrium model to describe this behavior. So we basically just added a layer to the MWC model that explicitly incorporates the dissipative phosphorylation-dephosphorylation cycle that we know is involved in this system. So we still have ligand binding degrees of freedom and receptor conformation or activity that controls on-off switching of this phosphorylation reaction. And then the key new insight is that the role of methylation isn't to modify one of the energies in this MWC Hamiltonian, but rather it lowers the energy barrier for this phosphorylation reaction. And because the system's driven out of equilibrium, lowering this energy barrier modifies the non-equilibrium steady state in a way that can produce this downstream shift in the response across a wide range of concentrations. But because it's implemented in this non-equilibrium way, there's no feedback to the ligand binding degrees of freedom. Okay, so that's a basic summary of our model, and I'll just show you the result fitting this main data set. We can now coherently fit both the downstream response and the wide spread in these response curves and the very narrow spread with methylation of the input response. So that's one story I wanted to tell you about. And with the remaining time, I want to tell you about a second experiment, which is coming from Tom Schmitzu's lab in the Netherlands. And they found this very surprising result that the signaling activity of E. coli spontaneously switches between fully active and fully inactive signaling states in the absence of any external stimuli. So there's no ligand involved here. And here's some examples of single cell trajectories that they measured that exhibit this switching behavior. So they took these switching trajectories and extracted various timescales. There's dwell timescales, how long the system spends in the active or inactive state. And there's also these transient or conditioned switching time scales, how long it takes to actually execute a switch between active and inactive. And here, the surprising result that reveals the non-equilibrium nature of the system is that the switching times are asymmetric. So it takes longer to execute the switch to the inactive state than vice versa. And here I'm showing the switching time distributions that they measured. So you can show quite generally that if detailed balance is satisfied by the underlying dynamics, these switching time scales should be identical. So again, this is a signature of the underlying dissipation in the system. So what we've done to understand this behavior is to build a non-equilibrium lattice model to try to reproduce this asymmetric switching. So here the nodes in the lattice represent what's called a core unit of the chemosensory array, involving several receptors and a kinase, which is the enzyme that does the phosphorylation reaction. And of course, this kinase is going through a non-equilibrium cycle that involves binding and phosphorylation of QI, the messenger protein. Between the kinase and the lattice, we have cooperative coupling. So active kinase tend to increase the activity of their neighbors. And we can also incorporate things like the external ligand concentration and methylation through something that looks like an external field, if you want to use an analogy to icing type models. Okay, so I'll show you some of the dynamics of this model and tell you about a couple other results. So with sufficiently strong coupling, we see spontaneous switching between active and inactive states in the system. And we can bias towards more active or less active by changing the external ligand concentration, for example. And then extracting timescales from these switching trajectories, the dwell times are exponentially distributed, which is what's seen in experiments. And most importantly, we reproduce this asymmetry in the forward to backward switching time because of the dissipative nature of these cycles in the lattice. So I don't have time to tell you about kind of all the investigations we've done with this model, but I'll give you some teasers. Comparing to the experimental timescales indicates to us that E. coli operates near a non-equilibrium critical point based on the relative timescales of switching, dwell time, and the asymmetry. We can also use things like transition state theory to understand the microscopic origins of this switching time asymmetry. The system's following different paths depending on the direction that it switches. And there's also interesting response behaviors of this lattice. We found a speed sensitivity trade-off in the response to external ligand concentration that's eased by increasing the dissipation within these nodes. Okay, so with that, I'll leave you with these concluding thoughts. So we have two non-equilibrium models that are able to capture these fundamentally non-equilibrium measurements of the chemosensory array. And we're also hopeful that these modeling frameworks can find broader applicability in two-component signaling systems and for studying things like non-equilibrium phase transitions. So if you're interested in the details, please check out our papers and I'll